Welcome back, everyone. We have made it to chapter 47. In chapter 47, we're going to talk about embryonic development. We're going to focus on animals, people, but also other animals. So, when we are talking about embryonic development among animals, there are a lot of common features of the early embryonic stages among different animals. In people, we can study a lot of embryonic development as far as like in vitro before it's been implanted. But otherwise, we really use a lot of model organisms to study development. A lot of those model organisms are used because they're easier to study in the laboratory rather than, making a pregnant woman stay in the lab for 40 weeks while you probe and test a baby. It's a lot easier to just use a model organism and then see how that reflects in different organisms and how it relates back to people. Development will occur at many points in the life cycle of an animal, so we're going to talk about a lot of embryonic development, but there's also development that can happen in animals after embryonic stages also. And we're going to see that across the range of animal species, embryonic development will have common stages that go in a common order. We're going to look at sea urchins. We'll look a lot at chicks. We'll look a lot at frogs. Those are a lot of the model organisms that we'll use. Sometimes flies or worms are used as a model organism. We won't really talk about those very much in this chapter, though. So in embryonic development, we kind of have the same cycle that occurs. First is fertilization, sperm and egg fertilized. That is the first thing that has to happen for anything to even develop. Following fertilization is cleavage. If you remember, we've talked about cleavage a little bit already. This is rapid cell division without growth. So you're going through, instead of the G1, S, G2 stages, you're just going through S, which is DNA synthesis, right into mitosis. The cells aren't growing. And you do that until you get to a ball of cells. And then that ball of cells that 16 cell stage will continue going through cleavage until you get through this hollow ball called the blastula. At that stage, gastrulation kicks in. Gastrulation allows some of these cells on this ball to start moving inward and forming layers. And so those cells are being rearranged into cell layers and cell tissues. We talked about some organisms are triploblastic with 3 layers and some are diploblastic with 2 layers. So at that point you have a gastrula. And then organogenesis is genesis meaning beginning, organo meaning organ. So you're forming your organs at that point. So the tissue layers are going to develop into actual organs. Here in this little frog, you can see you've got this embryo that's actually starting to get, form a tail and form its head. Eventually, that animal is going to hatch from its egg or be born, and growth and even metamorphosis can continue after that point. So in the frog, it goes through a larval stage when it's a tadpole to an adult stage. In people, it would be, a baby turns into a child, a teenager into an adult. And then that adult will be able to form gametes, whether it's sperm or eggs. Fertilization occurs, and the whole process repeats for the next generation. So we're going to talk about some important steps that happen to allow fertilization to occur. So to have fertilization, you have to have an egg and a sperm connect with each other. But it's also important that it is one egg with one sperm. If you have a bunch of sperm getting in, you'd end up with too much DNA, which is going to cause defects and likely even embryonic death. So we're going to talk about 2 types of reactions. One is the acrosomal reaction. If you remember the last chapter, we talked about the anatomy of a sperm. At the front tip of it is the acrosome. And then this is an egg, and you can see at the edge of it, it has a jelly coat. This is specifically talking about a sea urchin. So we're going to start with talking about a sea urchin model. But this is the egg with its jelly coat. And that jelly coat is just like this little siren call that is saying, oh, little sperm, come to me. And so the sperm kind of follow it, track it down. And the acrosome is able to interact with this jelly coat, and you have the acrosomal contents being exocytosed out. And that allows these hydrolytic enzymes to come in contact with the jelly coat. Hydrolytic means things that break down. So it's going to start creating reactions until you have the acrosomal process from the sperm fusing with receptors, with sperm binding receptors on the egg. This is an important step of speciation. So specific species will have different types of acrosomal processes or sperm binding receptors. So only, the sea urchin sperm can bind to the sea urchin egg. You're not going to have a frog mixing with a sea urchin or something. So you now have the sperm and the egg attached to each other. And the sperm and the egg plasma membranes are going to be able to fuse together. That's going to cause the sodium ions to come into the egg, and that is the acrosomal reaction. At that point, the egg will change to not be able to bind any sperm. So the acrosomal reaction gives you a very short term, but very quick. It takes about a couple of minutes, but only lasts a minute or two after that. Very quickly, that egg is sealed off to other sperm. Okay, what is the function of the acrosomal reaction in sea urchin eggs? This is going to cause the cortical reaction to occur. So in the egg, just under the membrane, are these little vesicles called cortical granules. And when the sperm and the egg membranes fuse, these cortical granules are going to spill their contents out to the edge of the egg, and that is going to trigger the fertilization envelope to occur. So this is going to cause these sperm receptors are going to be snipped off, and the fertilization envelope is in place, and the egg has a more slower, longer lasting block to having any additional sperm coming in. One thing that's interesting is the cortical reaction requires a lot of calcium. And so we're going to spend a little bit of time talking about calcium and how it affects the cortical reaction. Okay, how does a fertilization envelope form in sea urchins? And what is the function of the fertilization envelope? Okay, so the cortical reaction requires a rise in calcium ions. And you can follow through and see. So this is showing, each little spot is showing where the calcium ions are. So a second before fertilization, the spots are just kind of, there's plenty of them there, but they're all over the cell. But they're not clustered. They're not particularly bright anywhere. 10 seconds after fertilization, you can see a really bright spot. This is the spot where the sperm entered, and there's a lot of calcium right there. And that is also where this fertilization envelope is occurring. At 35 seconds, this wave of calcium has spread down about, throughout the sides of the cell. And you can see the fertilization envelope is continuing to be formed around the sides of the cell. At 30 seconds, the calcium wave is here at the other opposite pole of the egg cell, and the fertilization envelope is being formed here. Something that's nice is that these are rapid changes. The proteins and the mRNA that are going to be needed are already present in the egg before this whole process starts. The egg doesn't have to stop and create mRNA and proteins to allow this reaction to happen. It's just the egg fuses, the calcium is released, the fertilization envelope starts being formed. And at this point, cleavage begins. The cell is going to start going through cell division. Okay, we're going to watch the cortical granule reaction here.
Watch as cortical granules fuse with the egg cell plasma membrane immediately following fertilization by a sperm cell. These images were made using a lipophilic dye that binds to the membranes of the granules, allowing us to see individual fusion events. Notice how these fusion events, which are triggered by calcium, sweep across the egg surface immediately following sperm entry at the upper left. Credit: Mark Terasaki, University of Connecticut Health Center.
Okay, and let's follow calcium release.
See the changes in free calcium in an egg cell immediately following fertilization by a sperm cell. Simultaneous images of the fertilization event were taken using phase contrast microscopy to show the cell left and calcium green fluorescence microscopy to reveal areas of free calcium right. Notice how a wave of free calcium moves quickly across the cell, starting at the point of sperm entry in the upper right. Credit: Mark Terasaki, University of Connecticut Health Center.
So what would happen if you injected calcium ions into an unfertilized sea urchin egg? In mammals, let's talk about what happens in mammals. So the egg in a mammal is surrounded by these follicle cells. They're kind of like her little bridesmaids that are saying, we're going to keep you away until we approve of you. The sperm has to travel through the follicle cells, and then it reaches the zona pellucida, which is the extracellular matrix of an egg in a mammal. When the sperm binds, it triggers a cortical reaction similar to what we just saw. In mammals, this is going to be a lot slower. So the sea urchin, this whole process is about 90 minutes and it starts dividing. In mammals, it takes 12 to 36 hours after the sperm binds before you start the cleavage process occurring. So then we get to cleavage. So the fertilized egg has a single nucleus by this point. In some animals, actually, if we're talking about mammals, the sperm and the egg are still separate at this stage. But that fertilized egg is going to allow cleavage to occur. Cleavage is not going to increase the size, but it will increase the number of cells. And each cell will have the correct amount of DNA in it. So you have a haploid sperm and a haploid egg. To make a fertilized egg, it splits into two cells, DNA replication, cell division. You have two cells that are each diploid, then 4 cells that are diploid, 8 cells that are diploid. Eventually, you're going to create an early blastula, which is a ring of cells. In a mammal, you have a fertilized egg, which still has a haploid sperm nucleus and a haploid egg nucleus. You go through the first round of cleavage, and that is the point after the first round of cleavage that the two cells actually have one nucleus in each cell that is going to be diploid at that point. So we're going to go through 4, 8, and 16 cell stages when we're in a ball like this. And each of these individual cells is going to be called a blastomere. When it gets to the 16 cell stage, it's a nice solid ball. It's called a morula. Then around the 100 cell stage, this ball that's hollow in the middle is called a blastula. And inside is a fluid called a blastocele. That blastula continues going through cleavage, and you get a later blastula that you see is still about the same size but has more cells in it. In echinoderms, cleavage is going to be radial. It's going to be reducing in size. Echinoderms are uniform across the embryo. In frogs, there is an asymmetrical cell division. The different regions of the embryo are going to be a little bit different in the number and size of their newly formed cells. And a lot of that is because of the embryo yolk. So the yolk is a pack of stored nutrients in the egg for the embryo to use. And it's typically going to be concentrated towards one end. So wherever the yolk is concentrated is called the vegetal pole. So down here, the yolk is here. Up at the top is going to become the animal pole, and that's going to be contain the embryo. And so each pole has different colors and has impacts on further development. In frogs, the first cleavage is going to come right down the middle from pole to pole. So from the animal hemisphere to the vegetal hemisphere, just straight down. However, once it hits the yolk, that cytokinesis is going to be slowed down. So you're still making this first cleavage furrow by the time the second cleavage furrow begins. That second cleavage furrow just twists it on its axis, but it's still going from pole to pole, and will still slow down. That third cell division is more like equatorial, if you think on a globe, where the equator would be. However, the yolk kind of pushes that mitotic equipment away from it, so you end up with a smaller top, a smaller mass ending up in the animal hemisphere than in the vegetal hemisphere. It's also going to cause a blastocele to form entirely in the animal pole, because the yolk is taking all the space in the vegetal pole. Okay, now we have our blasts. Okay. In some animals, depending on how much yolk is there, this process is going to occur a little bit differently. Holoblastic cleavage is going to be complete division of the egg. This is what happens when there's little yolk or moderate amounts of yolk. So you end up with cleavage going through this kind of process, what we just saw in sea urchins and in frogs. Meriblastic cleavage is going to happen in animals with a lot of yolk. So this would be like reptiles, including birds, fish, and insects. If you think of a chicken egg, the yolk inside of it is actually like the entire embryo. The white doesn't turn into any of the animal. The yolk is the embryo, and most of that is that solid yellow yolk stuff, right? Only a little bit of it would actually turn into the animal if it were fertilized. In meriblastic cleavage, you have an incomplete division of the egg. So you end up with kind of this little like cap of cells growing on it. Sometimes, or typically, it'll turn into 2 layers of cells here. Okay, so once you have your blastula, you don't want your animal to just stay as a circle of cells. So we're going to start morphogenesis. Morphogenesis is the process that lets an animal's body take shape. Morpho means shape, so we're giving a shape to this. This is going to be the last 2 stages of embryonic development, gastrulation and organogenesis. Gastrulation is when the cells at the surface of the blastula are going to move into interior cell layers, and it starts forming the primitive digestive tube. So here you're starting to get the digestive system forming here. Organogenesis is when you are forming the organs. Gastrulation will reorganize the blastula into either a 2-layered embryo or a 3-layered embryo. And this stage is called the gastrula. Only animals gastrulate. This isn't something we see in plants or fungi. And you're going to have cell movement throughout these processes, and that will form layers. And these cell layers are called germ layers. That word germ refers to like germinate, growing. So this is, how the embryo is growing. Eventually, the late gastrulation stages, you will have the ectoderm that are going to form the outer layer, the endoderm in the inner layer, and if it is a 3-layered embryo, that middle layer is called the mesoderm. Organisms with 3 layers are triploblastic, and organisms with 2 layers are diploblastic. Gastrulation starts with this process called invagination. That's what's going on here. It's invaginating in. Folding in these cells. And this is going to allow germ layers to be produced, and those germ layers will give rise to various tissues in our body. The 3 germ layers we have are the ectoderm, the mesoderm, and the endoderm, and they each have specific jobs for what they end up creating, although some parts of your body, like the adrenal gland, comes from here and here, so not everything is only one thing. The ectoderm is going to be the outer layer of the embryo at this stage, and those cells are going to give rise to the outer layer of your body. That would be the epidermis, the skin and skin's derivatives, so sweat glands, hair follicles. It'll also give rise to your nervous system, your sensory systems, your pituitary gland, adrenal medulla, so a lot of your glands, your jaws, and your teeth. The mesoderm, or the middle layer, is going to give rise to your bones and muscles, your skeletal and muscular system, your vascular systems, which are your circulatory and lymphatic systems, your dermal tissues, and the inner parts of some glands, so your excretory and reproductive systems, the dermis of your skin, and the adrenal cortex. And then your endoderm is going to give rise to the cells that line your body systems, so your epithelial lining of your digestive tract and the organs of your digestive tract, the epithelial lining of your respiratory, excretory, and reproductive tracts and ducts. And it'll also give rise to some of the glands of your body, your thymus, your thyroid, and your parathyroid glands. Okay, here's another embryonic development.
The process of gastrulation rearranges the single layer of cells in the blastula to form a three-layered gastrula. Cells at the vegetal pole invaginate, move inward, forming an indentation called the blastopore. The embryo is now called a gastrula. Around the blastopore, mesenchyme cells break away and migrate into the blastocele. The indentation that started at the blastopore becomes a tube called the archenteron, and it will eventually form the sea urchin's digestive tract. Mesenchyme cells pull the archenteron toward the animal pole. The blastopore will become the anus, and the archenteron will break through near the animal pole to become the mouth. Gastrulation is complete when the embryo forms three tissue layers: ectoderm, endoderm, and mesoderm. The ectoderm is the outer layer of the embryo. During the subsequent process of organogenesis in vertebrates, it develops into the outer layer of skin and the nervous system. Endoderm lines the rudimentary digestive tract of the gastrula. In vertebrates, it eventually forms the lining of the stomach and intestine, and digestive glands such as the liver and pancreas. In between these two layers lies the mesoderm, which started with the mesenchyme cells and develops into most remaining organs and tissues, such as the skeleton, muscles, and circulatory system.
Okay, in a frog embryo, gastrulation is going to start. Well, first let's remember a frog has a front and a back, a left and a right side, a top and a bottom, right? It's has bilateral symmetry to it. And so we're going to start getting some front and back and all of that in our embryo. So gastrulation in a frog embryo begins when cells on the dorsal side of the embryo start invaginating to form the blastopore. You get a dorsal lip. So I think of like if you purse your two lips together, the like right where your lips touch is that little crease. That's like what the dorsal lip is. So the dorsal lip is the cells right above the blastopore. These cells that moved inside are going to become the endoderm and the endoderm and the mesoderm. The cells on the outside are going to keep moving. They're going to keep changing shape. Then the blastopore is going to start moving in on both sides. And this blastocele is going to be gradually shrinking. And the archenteron is going to start forming. The archenteron is going to become the gut of the frog. And the blastopore is going to become the anus. Then late in gastrulation, the cells that are remaining outside will be the ectoderm. The endoderm is the middle layer, and the mesoderm is, the endoderm is the inner layer, and the mesoderm is the middle layer. And then the blastopore opens to the archenteron, and the blastopore will fill with a yolk plug, which are yolk cells. In chicks, if you remember, a few slides earlier, we talked about they're going through this meriblastic cleavage where they have this little cap on top. Okay. In chicks, so it's just this little layer on top that we're talking about. So this embryo is bilaminar. There's an upper layer, the epiblast, and a lower layer, the hypoblast. And this all lays on top of the yolk sac. So if you were looking at like a chicken egg that had been fertilized, not just like from the grocery store, those are not fertilized, but one that is fertilized has a yolk and then just little tip of it here. This is what the cells that we're talking about here. So this is the embryo. And the cells that are going to produce the embryo are coming from the epiblast. Some of these cells move downward, and those are going to form the endoderm. And some of them move laterally, that's going to be across the side. They're going to form the mesoderm. During gastrulation, the epiblast cells are going to migrate toward the midline of the blastoderm. You can see these arrows showing they're moving this way. And then into the embryo toward the yolk. The midline is going to thicken, and this is called the primitive streak. And the hypoblast cells will contribute to the sac that surrounds the connection, that surrounds the yolk, and the connection between the embryo and the yolk. But these are not going to contribute to the embryo itself. Okay, we're going to pick up with talking about gastrulation in humans and finish the chapter in our second part of our video. I will see you soon. If you have any questions, let me know. I hope everyone's doing great.